what are the things to be considered, even if you're planning to buy a machine, or if you bought a machine, what to do next, and what are the precautions has to be done with respect to technical and non-technical both, right? So the microbes, it's not in one place. We all know it's there anywhere and everywhere you name it. Be it an industry, like say you walk, wherever you walk, be it a park, theater, school, college, or office, everywhere it is there, right? So there is no, we are living in microbe world rather than they living in our world. That's what we see, right? And how do we understand? Is it easy to understand? So since we are seeing it almost everywhere and anywhere, right? What is the possibility to understand it in a better form, right? Of course, the microbiology term is not something new, but once the genomics comes in picture, the way we started to look into microbiology is very, very different, right? Because we came to know many new things and how it has been done apart from the normal traditional microbiology test. When we do NGS specifically, what are the things we are going to get and what are the equipments which will be using, we are going to see that, right? So there are two kind of sequencers generally in market, right? One is a benchtop, another one is a probably the production based. So when I say benchtop, what does it can do? Benchtops are made for specifically smaller organism sequencings. So as you can see the list in this, right? You get a pointer. So these are all the applications, these are all the machines, right? So most of the, not only in India, across the globe, if people are having limited sample, be it clinical or non-clinical, they are mostly starting with MySeq. What is MySeq is basically, it's a small benchtop which allows you to produce minimum of 15 GB output. When I say GB, it's gigabases here. So as far as the wet lab is concerned, you will never ever speak about the bytes, which is a computer term, right? So the total output which we get from the machine is basically 15 gigabases, right? So again, every machine will be having their own set of specifications in specific. So if I use this machine, how many samples I can load, how much depth I will be going, how many probably, what are the things I can understand with respect to application also. Suppose example here, as you can see here, yes, right. So here, these are all the applications, right? So as you can see, these dots represents what are the application we can do with respect to machines. So broader machines can do anything, anything and everything. When I say anything and everything, it means including your smaller organisms, be it a fungus, virus, bacterial, or any kind of microbes, and the humans. But when we say benchtops, humans, you can do only targeted panels, but microbes you can do, like even if it is a less number of samples, even the benchtop will solve the problem. So in that case, MySeq is mostly preferred for small whole genome sequencing of microbes and viral, right? So of course you can do everything. It's based on the sample load, but the optimized size, when you say not so less or not so more, which is something average or the mid-level, MySeq is preferred, right? So again, you can see, so what are the applications generally used from the microbes world? Either it is a whole genome or probably even 16S metagenomics and or the full, like the shotgun metagenomics or meta-transcriptomics, right? So these are all things generally done. As far as metagenomics is concerned, if the sample load is lesser, then that can be done in MySeq. If it is more, then the bigger machines are involved. So when I say, so why the more and less comes in picture very often? Because the costing is involved, right? So how do people plan the project or probably study the total, like say, if you have a data set or if you're planning to study something, first thing will be estimating how many samples you are going to process, how many controls are going to be there, how many like say in your case, if it is a time series data sets or if it's a control versus normal, you'll be planning everything, which machine fits in, which flow cell goes in, what are the reagents has to be purchased, everything should be planned forehand, right? For that, this table will be useful. It's like an easy, probably the reference. This is for the benchtops.